Hello everyone, my name is Rizzo, and today for Honors Day I will be presenting a presentation titled The DNA Methylation Signature of EpCAM negative CD49 negative breast cancer stem cells correspond with worse clinical severity in basal-like breast cancers. So this research has been done with Dr. Austin Scholl. I began this in 2020, so we've been doing it for about 2.5 years now. So to start things off, in this study, we explored breast cancer prognosis and how certain breast cancers are more prone to relapse and metastasis, which is important because we have seen that when there is relapse and metastasis, there is a big decrease in survival. Therefore, we can try to understand some of the mechanisms behind this difference. So cells in a breast cancer tumor are actually very heterogeneous. There are different types of cells within a tumor population, and some of these types of cells actually serve as a source for other types of cells. So they basically turn into different cells in the tumor, and these are known as cancer stem cells. They're typically more resistant and more prone to progression, relapse, and metastasis, and therefore we want to better understand cancer stem cells and how they correspond to worse prognosis, because hopefully in the future we can then better identify them and eventually target them in a clinical setting. So on this slide, you can see 3 different types of breast cancer tumors. So tumors are made up of different types of cells, and you have more mesenchymal, which is more of the stem cell type of cells, and then you have differentiated cells, and you can see that the mesenchymal cells really make up basal tumors, which is more of your aggressive cancers like triple negative breast cancer, for example, whereas your luminal cancers are a little more differentiated in their cells and have higher populations of those differentiated cells as opposed to stem cells. And those are typically easier to treat. As you can see, this little line here at the bottom, you have the best and worst prognosis. The worst prognosis goes with those basal cancers that have a lot more cancer stem cells than differentiated cells. So on this next slide, you can see 2 different graphs, one of relapse and one of distal metastasis. So relapse is after you've had cancer and had treatment and it's starting to get better. You have a relapse, or you have a new occurrence of cancer. Your cancer comes back. And metastasis is when your cancer spreads into different organs in different parts of the body. So you'll notice that this red line is the cancer stem cell populations in those more aggressive cancers, and they have a higher incidence of relapse and metastasis. So here you can see in this circle, you have these blue stem cells, which eventually differentiate into luminal mammary tissue. And right next to it, you'll see this little picture where the blue cells are the stem cells. And you can see that typically normal therapies will target the more luminal differentiated cells, and that leaves this area for the cancer stem cells to rapidly grow and metastasize, where if you could find a way to target the cancer stem cells, those blue cells on the outside, it would lead to regression. So because we're trying to understand the development of cancer cells, we have to look at the typical cellular process of epigenetics, which is a process that modifies DNA in a way that it expresses certain genes within certain cell types. And this process doesn't actually affect the sequence of DNA. It's just certain alterations that cause different genes to be expressed differently. So 2 of the most common types of epigenetic modifications are histone modification and DNA methylation. So histone modification is where your strand of DNA becomes wrapped around a histone protein, which prevents transcription. And you also have DNA methylation, which is a CH3 or methyl group that gets added on to different spots on the DNA, typically cytosines and guanines. And these can alter the way that certain genes are expressed. This project specifically focuses on DNA methylation. So we wanted to take the approach of understanding what the DNA methylation patterns are in cancer stem cells and how they differ from those in non-cancer stem cells. So first we had to determine our cancer stem cell and non-cancer stem cell populations. So flow cytometry was used with EpCAM and CD49F, which are actually epithelial markers on the cell surface that show that the cancer stem cells have been differentiated. So you have this group over here that are double positives, and we use those as our non-cancer stem cell population, and those who are negative, showing that they have not been differentiated by either one, serve as our non-cancer stem cell populations because they display neither of those differentiation markers. So in some previous work with collaborators, we isolated the cancer stem cells from the non-cancer stem cells from a culture of a mixed population of cells. We then isolated the DNA and performed a 450K DNA methylation analysis, in which we were able to look at 450,000 DNA methylation sites within the genome. You'll notice that on this heat map over here, you have a very distinct division between this little stripe here on the left and the rest of it. And so this stripe on the left is actually our cancer stem cell population that we isolated. So next we just kind of confirmed that the group that we chose as our cancer stem cells that were negative for both of the epithelial markers are actually linked to those basal-like breast cancers, just like we predicted. So you can see that the populations that have very high numbers of those negative for EpCAM and CD49F are actually basal breast cancers, which is shown here in the orange, whereas some of the populations that have pretty low of those what we consider cancer stem cells are either luminal or normal. Next, we did a principal component analysis, which uses a lot of different factors to separate data into groups, and you can see that our cancer stem cell rich populations are separate from the non-cancer stem cell populations in a lot of different ways, and we separated these into two different groups, the CpG island promoters and the non-CpG island promoters. So before, when I was talking about methylation, and I was saying that these methyl groups tend to attached to cytosines and guanines, well, a CpG site is a site where there is a cytosine followed by a guanine, and it's typically a spot where methylation occurs. So you have these CpG islands, which are kind of concentrated locations of these CpG sites and promoters, and then you have non-CpG islands where there is not a significant amount of CpG sites in the promoters. But in both CpG island and non-CpG island promoters, you can see a difference between the cancer stem cell rich and the cancer stem cell poor populations. So here we have the heat maps that are showing the difference between the cancer stem cell and the non-cancer stem cell in CpG islands and non-CpG islands. So this area that has the little boxes around them are our cancer stem cell populations. So you can see that in CpG island promoters, most of the genes are hypomethylated, but you have the cancer stem cells that are hypermethylated. And then if you go over to the non-CpG islands, you will see the opposite. You have hypermethylation in the regular cells, and then in cancer stem cell populations, you have hypomethylation. It's also interesting to note that each of the cancer stem cell rich cell lines contains a genetic alteration in the TP53 pathway, which is a tumor suppressor, so that makes sense. And then the PI3K-AKT pathway, which is kind of concerned with growth and regulation and differentiation and things like that. So that also makes sense. So now if you look at some of the biological processes that are in CpG island promoters, you have differentiation and development and things like that, which makes sense because these are hypermethylated in cancer stem cells, and they remain in that stem cell mesenchymal state. And then in the non-CpG island promoters, you have things like defense, inflammatory response, immune response, and things like that. And those are hypomethylated in cancer stem cells. So then what we did is we looked for the genes that were differentially methylated in CpG islands between the cancer stem cells and non-cancer stem cells, and we got around 2,100 genes. Then we looked for genes that were differentially methylated cancer stem cells versus non-cancer stem cells in non-CpG islands and found about 1,000 genes. So we took the overlap between those two, looking for genes that were differentially methylated in both CpG islands and non-CpG islands. So that left us with 232 genes of interest. So then we used TCGA data, and we were able to look at actual clinical data. And of those genes that were differentially methylated, we saw which ones corresponded with worse progression-free survival and worse overall survival. So progression-free survival would be you don't have progression in that cancer. It doesn't get worse and you do survive. And then overall survival is just the overall survival of the patient. So 60 of those genes corresponded with worse progression-free survival and 44 corresponded with worse overall survival. So again, we took the overlap between the two. We found 27 genes that were differentially methylated in both populations that were associated with both worse progression-free survival and both worse overall survival. So of those 27 genes, 4 of them listed here are OVA1, DOX7, RBM47, and PLEC-HA7. So OVA1 is a transcriptional repressor, DOX7 is involved in the neuromuscular junction, RBM47 enables RNA binding, and PLEC-HA7 is involved in epithelial cell-to-cell adhesion. So in all 4 of these genes, this worse survival occurs when the methylation rates are lower. And interestingly enough, all 4 of these genes are expressed lower in basal breast cancers as well as in triple-negative breast cancers, which are typically associated with worse prognosis and metastasis, which is a characteristic that we described of cancer stem cells. So something that we've been looking at this semester specifically is looking at these 2 distinct groups or populations in different types of cancers and not just breast cancer. So we tried to focus on epithelial type cancers. So we looked at cervical cancer, glioblastoma, and colon cancer, and we were just kind of getting our feet wet, seeing if there were 2 distinct visible groups, which we did see in all 3. You can see there's a clear difference between one group and the other. So to try to kind of confirm whether or not this could be a good predictor of more aggressive cancers in these different cancer populations like it is in breast cancer, we looked at the data for overall survival, and there was no clinical correlation of overall survival with the populations that look more similar to the cancer stem cells. We did use non-CPG islands for these heat maps, so it would be interesting to repeat the process with CPG islands, and it would also be interesting in the future. Hopefully some of Dr. Scholl's later research students will be able to look at things other than overall survival, so maybe age of diagnosis or whether or not there was progression or things like that. So hopefully they would be able to find a way to use these differential populations as predictors for more aggressive cancers in these other cell types. So in conclusion, there are differential methylation patterns between cancer stem cells and non-cancer stem cells. Genes containing cancer stem cell specific DNA methylation changes also contain differences within the breast cancer patients from the TCGA data. And cancer stem cell associated changes in patient data correspond with worse survival, further highlighting the correlation between the presence of cancer stem cells and clinical severity. So some future directions include investigating whether or not the cancer stem cell or non-cancer stem cell methylation markers could be predictors of worse prognosis and other types of cancer. So this is what we kind of started right at the end with the cervical, colon, glioblastoma. However, this could also be investigated in pretty much any other kind of epithelial cancer where there is differentiation from a stem cell to a differentiated epithelial cell. And then also when observing CPG versus non-CPG island methylation patterns, is one more predictive than the other for worse prognosis in breast cancer? Looking overall big picture, we've been thinking that non-CPG islands are a better predictor, which is why that is what we used for those heat maps at the very end. However, there could be more research kind of confirming that non-CPG islands would be more predictive than CPG islands. And finally, how could genetic screening look at differential methylation as a predictor of worse prognosis in a clinical setting? So basically, the end goal would be that you would be able to genetically screen somebody, and if they had these markers, these methylation markers, you could show that they are more susceptible to having a more severe type of cancer as opposed to a more differentiated, luminal, easier to treat type of cancer. For my acknowledgements, I would like to thank Dr. Austin Schull for putting up with me for the past 2.5 years. Lainey Benson, me and Lainey have kind of worked side by side doing kind of parallel research this entire time, and a lot of the work that I built my research off of was previous work by Emma Gray, who's now in medical school. We have a lot of collaborators from all over the world, and we've received funding from the South Carolina Embry program. I also was fortunate enough to get to present this information at the South Carolina Embry Idea Research Conference, the South Carolina State Conference, and I also was able to present it at the American Society of Biochemistry and Molecular Biology Experimental Biology National Conference. So that was really cool. I got to present this in sort of a poster format and do the undergraduate poster competition. And this is also my third honors day that I have been able to present this research at. We basically have added a little bit of information and a little bit of research every time, and this is kind of the culmination of my 2 years of work. And finally, I would like to dedicate my presentation to two of my sorority sisters, Haley Hughes and Morgan Megna, who both passed away after their battles with childhood cancer last fall. Thank you.